Well, okay. Uh, Thank you for coming. I'm pleased to introduce William Wiefer today. He's a PhD candidate in ecology and evolutionary biology at the University of Michigan in Ann Arbor since 2019. And he is promising his advisors to finish within the next 5 or 6 months. And he got his BE in ecology and evolutionary biology and one in environmental studies in 2017. Today, his talk is entitled AI Meets Nature, Expanding Access, well, I can read from here, Expanding Access, Utility and Discovery in Natural History Collections. We are very interested in your talk, and without further ado, can I ask you to start? Thank you.
Well, good afternoon. Thank you all for coming. I just wanted to thank Matt, Torsten, and Todd for helping get this set up. It's really exciting to be here. So first, I would like to invite you to take a little journey with me away from Chicago's chaos. We're out in the field, deep in Michigan forest, surrounded by towering oaks and sycamore trees. The air is crisp, and the ground beneath our boots is soft with all of these falling leaves. And as biologists, we're here on a mission. We're here to study the incredible biodiversity of this forest. We've got our trusty field notebook in our hand. We're ready to capture all of the things that we observe. We record the size of the trees, the species we spot. We might even sketch a few leaves. And the weather matters, so we take note of that as well. And with our camera, we document our surroundings, knowing that that will help us interpret our results once we get back to the lab. And of course, we collect samples. We collect leaves, twigs, fruits, seeds. Each of these items is a small piece of this forest story. So after a long day of field work, we head back to the lab where the real detective work begins. We analyze our notes, crunch the data. We might even extract some DNA to help better understand the forest's genetic composition. And eventually we'll write all of this up in a manuscript and hopefully it'll get published. That's the way these things usually go. But there's more we can do. What if we want to make sure that our data has a lasting impact? What if we want to make sure that our data goes beyond just this manuscript, beyond one journal article? We can do something extra, something that reaches beyond the present. We can create a specimen voucher. So what is a specimen voucher? It's a physical record. It's proof that an organism existed at a certain place at a certain time. And in our example, we would dry and press flat a small branch or a few leaves from each of the trees we encountered. And document all of the important information on that specimen's label. We can then deposit this specimen into a museum, or more specifically an herbarium collection, where it'll be stored safely for the long term and available to other researchers. So these are some fun examples of herbarium specimens. And on the left is a pretty ornate coffee specimen from 1780. And in the middle is a variety of teas from 1841. And then on the far right is Liquidambar styraciflua, which is sweet gum, and that's just one of my favorite plants. So if our Michigan forest specimens were accepted and accessioned into an herbarium collection, there's a good chance they would end up in one of these cabinets, tucked away in a protective folder, organized by taxonomy, much like the stacks at a library. Once our specimens become part of an herbarium collection, they become part of something bigger. It's not just our research anymore. It's a building block for future discoveries. Someone studying the impact of climate change decades from now might rely on our specimens to see how Michigan's forests have adapted. So while our fieldwork might help us answer some questions today, our specimen vouchers give our research a life far beyond its own. And that ensures that the story of this forest continues to be told. So that's all very exciting, but there's always a catch. How easy is it to access these specimens? And the answer is not very. If a researcher in California wanted to look at our specimens, they would either have to travel to Michigan or request a loan. And if you request a loan, there's always a risk of something happening to the specimen. So that's why in herbarium collections, we've been working for the past few decades to start to digitize our collections. By scanning the specimens and uploading their data to portals, we can create high-resolution digital records that are available to anyone. So now instead of waiting for weeks for a specimen to arrive, you can query a database. And if you do have to travel to see the specimen in person, you at least know what you're going to see. The largest of these data portals is called GBIF, and on it you can find just about everything. And suddenly we have a completely different problem. Now we have access to so much data that it's actually challenging to use it effectively. And that will be the focus of today's talk. And the title sort of gives it away, but AI and machine learning is capable of doing a lot of the heavy lifting. This is just a very broad overview about what we'll be talking about today. So we'll start with a little botanic background, some of the reasons I became interested in all of this, and then we'll talk about two specific tools I've been developing. One is called Leaf Machine 2, which is extracting trait data from herbarium specimens, and the other one is Voucher Vision, which is focused on extracting information from text-based resources. So we're going to move away from our Michigan forest example and dig a little bit deeper into some of the questions that I think about every day. Questions that I think require a massive amount of trait data to get a satisfying answer. So some of these questions are like, why do angiosperms have such incredible leaf shape diversity? When we go out into a forest, especially a temperate hardwood forest, every leaf is different. Why is that the case? Are there fundamental constraints determining these shapes? And then ultimately, what is leaf shape? Is it an adaptive trait? Is it the result of phylogenetic inertia? What constraints are governing all of these shapes? So this is a phylogeny of Vertiplantae, or green plants. Near the bottom, we have green algae, and at the top, we have angiosperms. And green plants are not evenly distributed across the phylogeny. For example, there are roughly 1,000 extant gymnosperm taxa, but there are perhaps as many as 350,000 angiosperm species. So the impact of angiosperms on biodiversity is truly astonishing. Darwin famously remarked that the origin of angiosperms is an abominable mystery. And that's still largely true. It's still complicated to figure out why there is so much angiosperm diversity. And that's where we're going to focus today. So studying leaves specifically dates all the way back to the Victorian period. And unsurprisingly, Darwin himself was a fervent botanist, and more of his time was spent studying plants than any other system. And the incredible diversity of plants and their representation in the fossil record spurred a lot of Darwin's research. He rigorously tested his theories by breeding and crossing plants in his gardens. Another early botanist, Johann Wolfgang von Goethe, also discovered that leaves were essentially building blocks for plants. So many features within plants are based on leaf architecture, such as flower petals. So even though the vertebrate anatomist, Sir Richard Owens, is credited with coining the term homology, von Goethe realized that many traits of related species could be transformed to fit a different purpose. So while not all of von Goethe's ideas have held up, he was correct about the serial homology of leaves. So in biology, homology has a specific definition. So biological homology is similarity due to shared ancestry between a pair of structures or a pair of genes. A common example given is vertebrate forelimbs, where they have homologous bone structures, but they are very different in their shapes and uses. So moving back to leaves, I don't know anything about mammals, so here we go. On this diagram, this kind of outlines some of the basic leaf shapes that we might be interested in. On the bottom, we have dentate and serrate margins, which are really common in temperate forests. At the top, you see an entire smooth margin that shows up just about everywhere in the world. And then on the left side, we have some more complicated architectures like sinuses and lobes. Oaks are the primary example of those. And then we have leaflets. So of course, some plants can't make things easy for us. Simple leaves are what we normally think of as a leaf, but all three of these photos only have one leaf in them. And we also have pinnate, we have compound leaves, and they come in all sorts of shapes and sizes just to make things even more complicated. Those are all fairly typical leaves, but each of these taxa have highly modified leaves that do all sorts of crazy things. So Sarracenia and Nepenthes are pitcher plants. Their leaves have been modified to consume insects to get more nutrition. And then we have cacti who have modified leaves into spines for protection or shading. And then on the right, we have Gloriosa. They have modified leaves. They become tendrils and help the plant climb vertically. So now that we have a little bit of anatomy background, we can take a look at some leaf shape trends. So some plant clades or groups of closely related species have virtually identical leaf shapes. And oftentimes these shapes are associated with environmental conditions. So looking at this Michigan forest photo from the fall, you'll notice that many of these leaves have non-entire margins. They have lobes, they have serrations, they have leaf teeth, and they come in a wide variety of shapes. But there are exceptions to this. So pawpaw is from the family Annanaceae, which is primarily a tropical family. Similarly, we have spicebush, which is from the family Leraceae, which is also a predominantly tropical plant. And then if you were to look up through a rainforest canopy, you might see something like this. And you'll notice that many of these leaves have entire smooth margins. They tend to be ovate and more rounded. And this pattern is a long-standing observation. In 1916, Bailey and Sinnott described the tendency of angiosperm leaves to have non-entire margins in temperate climates, like in Michigan, while tropical plants tended to have smooth and entire margins. And they also denoted that just how unreliable leaf shape was at determining species relatedness and common ancestry. Even species within the same genus can have vastly different leaf shapes. For example, these are sister species in the genus Begonia, and they share most floral and vegetative traits, but they have completely different leaf shapes. The biggest difference is on the left, Europhylla has entire peltate leaves, so the stem is in the middle of the leaf. And on the right, we have Heraclifolia, which has highly dissected, nearly compound leaves. Alternatively, some sister species have nearly identical leaves, while other traits vary. A good example of this is rhododendron, which have famously high floral trait diversity, but nearly homogeneous leaf traits. So leaf shape can be influenced by evolutionary history, but there's a lot of different factors that go into this. So this diagram also does an excellent job of showing just how much variation can occur in a single individual. These are red oak leaves from the same tree. And depending on the lighting conditions, the time of year, you can get incredible diversity within a single individual, let alone the entire species. So ostensibly, all of these shape differences can be quantified. There are three traditional ways of characterizing leaf shape. Traditional morphometrics rely on basic measurements that someone could perform with like a ruler and a grid sheet, things like leaf area, perimeter, and aspect ratio. And those are very important traits, but they definitely do not take into account the full complexity of a leaf shape. An alternative might be geometric morphometrics, which are computationally derived and rely on placing homologous landmarks on the leaf architecture. So this figure is of a geometric morphometrics approach implemented in the software MASS. And here they're fitting elliptic Fourier descriptors to the leaf shape outline, defining a curve, and then using that information to calculate a mean using Procrustes analysis. But whether we use elliptic Fourier descriptors or landmarks, we do run into a really big problem. And that's that landmarks require homologous structures, which we don't have across angiosperms. And then also comparing elliptic Fourier descriptors similarly doesn't work when you extrapolate across a large scale. So we need a method that allows us to compare disparate shapes in a unified morphospace. Another difficulty in shape analysis is how to summarize all of this variation. Means can oversimplify our variation. Intraspecific variation might even produce multimodal shapes that you would lose with a lot of summarization techniques. So we also need to develop methods that better take into account the inherent variation in all of these taxa. And then finally, we have the issue of scale. So many studies approach the issue of leaf shape from either end. They're either a cladistic study that has a lot of resolution, which provides a lot of insight, but then you can't make very many claims about the larger macroevolutionary picture. And on the other hand, you have studies that have looked at shape differences across higher taxa, so at the family or order level. And in this case, that tends to underestimate the number of transitions. So in either case, they also use binary data typically, so the presence or absence of a feature. And if you try to use continuous characters, you run into the problem of comparing disparate morphospaces. So this is a phylogeny from Givnish and Kriebel in 2017, and they looked at the evolution of leaf teeth across angiosperms using binary trait data. And if we focus on just the order Dipsacales, we can see how this study can be a little bit misleading. We zoom in on this red portion, that's the order level phylogeny of Dipsacales. And on the left, we have a viburnum phylogeny, which is a genus within Dipsacales. And in this case, black teeth means that there's no leaf teeth present. White means leaf teeth are present. So we can see in the Dipsacales phylogeny at the order level, it shows 2 transitions from non-toothed to toothed. But in the viburnum phylogeny, there's between 8 and 14 transitions. So ideally, if we want to study the macroevolutionary patterns of leaf shape, we would want to have the resolution of a genus level phylogeny like this viburnum study, but hopefully be able to look at it to study heterogeneity at a much larger scale like the Dipsacales project. So what tools do we need to begin chipping away at some of these questions? Well, first of all, we need a very large phylogeny if we're going to be doing a macroevolutionary study, which in itself is non-trivial. And secondly, we need a huge amount of trait data. And that's likely going to come in the form of millions of leaf measurements. And today I'll be talking about how I'm using Leaf Machine 2 to do just that. And then finally, we need a common morphospace, a way to incorporate all of these shape measurements in a unified morphospace where we can compare disparate shapes. So since the questions I'm interested in require so much data, for the last, at this point, 8 years, I've been developing Leaf Machine 2, which is an ensemble of 7 different machine learning algorithms that are designed to extract as much quantitative trait information as we can from herbarium specimens. So this diagram outlines the basic architecture of Leaf Machine 2. Each box represents a distinct machine learning algorithm. Green are object detection algorithms, cyan are large language models, gray is scene detection, orange is a key point detection system, and then white are instance segmentation. And the output from all of these models is aggregated together, and in turn, we produce a huge amount of derived trait data and really insightful archival metadata. Over the years, we've had to manually generate a huge amount of ground truth training data to make all of this possible. So for at least supervised machine learning algorithms, you have to show the algorithm the truth. And in that case, that means placing bounding boxes or points or tracing outlines of leaves in all of our herbarium sheets. So to do this, we've placed more than 500,000 individual bounding boxes over plant components and well over 200,000 bounding boxes over archival components. And similarly, we've placed basically 250,000 landmark points, and we've manually traced more than 30,000 individual leaves, bringing us well over 1 million manually placed landmarks, which is pretty crazy when I think about it. And we still have more on the way in the pipeline. And all of this has allowed us to do some pretty cool things. So I wanted to thank all of these folks for helping make that possible. Clearly generating 1 million ground truth measurements is not easy, and it's taken us probably 4 or 5 years to get there. So they've done a lot of hard work on this. This is a more visual outline of what Leaf Machine 2 is actually doing. Not all of the steps are included here, but we can cover the most important ones today. And the first thing you'll notice is that our component detection algorithm, they both drive the entire process. So we have a plant component detector and an archival component detector. And both of these are responsible for creating bite-sized pieces that we can then analyze with component-specific analyses. So to demonstrate that, let's follow this specimen through Leaf Machine 2. We start by identifying 11 different plant components, and these are simply bounding boxes around things of interest. So things like ideal leaves, partial leaves, buds, leaflets, flowers, roots. And these bounding boxes isolate those, and we only then process the cropped portion of those images. Once we isolate something like an ideal leaf, we can send that to be processed by our instance segmentation and landmarking algorithms, which looks something like this. We currently use Detectron 2 and PointRend to do the actual segmentation. And that finds the lamina, the petiole, and holes that might exist in the leaf. And since this whole process is modular, we have a lot of control over which leaves actually get segmented. You can imagine on an herbarium sheet, a lot of the leaves that are present are going to be partial, they're going to be incomplete, and we have a lot of control over how we actually process them. We also detect 11 different landmarks to get another set of measurements, and that includes tracing the petiole, tracing the midvein, measuring apex and base angles, things like that. And also, so stepping back a little bit, we can plot all of that information onto a nice summary overlay image to get a better sense of just how much data we're pulling out of the specimen. Here, I also wanted to show a little bit of Leaf Machine's segmentation ability. So Leaf Machine can generate high resolution masks, even in difficult scenarios and with difficult, complex leaf shapes. We also trained our algorithm to just ignore mounting strips. Plenty of herbaria use mounting tape to hold the specimen onto the sheet. Using traditional approaches, you would have had to reconstruct all of those leaf shapes. But in our data set, we just taught it to ignore it. So we can get really reliable leaf shapes, even if the leaf has been bisected in some way. Even though our algorithms are pretty good, the whole point of this project is to scale up trait-based projects. So we've also been working on automated quality control tools using visual question answering large language models. And we've developed a few different ways of doing this, but essentially we show the model one of our quality control images, give it some instructions and a rubric, and it will look at the mask relative to the leaf and assign it a score and provide us a reason for that score. So here you can see the visual question answering model, the VQA model, provide a score of 9 and says that there's a minor mask omission, which we can see it missed the very tip of the leaf. In this case, the bounding box cut off the very top of the leaf, so it assigns a slightly lower score. And depending on our requirements, we could either accept that measurement or reject it. And if there's no mask present at all, it acknowledges that too, and we can keep going. In our last publication, we tested Leaf Machine 2 across all angiosperm families and found acceptable performance in 245 out of 341 families. And it's hard to see from this, but a majority of the families that it does a poor job on, we would expect it not to do well on, something like cactus or angiosperms that have no broadleaves at all. So this has been pretty exciting. And this also informed the next stage of our ground truth training data preparation. Since this publication, we went back and have started annotating many more specimens from poor performing taxa, which has again increased our overall performance. So now I just wanted to give a sneak peek at some of our early results from applying Leaf Machine at a very large scale. And this has been able to happen because of Dan Chitwood and Elizabeth Munch at Michigan State University. And we've all been working on some methods to get us to the point of having the ability to compare all of these leaf shapes in a unified morphospace. And that's what this fun diagram is. We're able to compare all of the different leaf shapes that are possible in one unified morphospace, which is really exciting. And I won't be talking about those methods today, but in the near future, there's going to be some really exciting work about this. And when I applied to grad school, I had the goal of measuring all angiosperm leaves. And luckily, my advisor, Stephen Smith, did not laugh me out of the room. And happily, that's almost a reality now. A week ago, I had 0 leaf measurements for the orders Corneales and Magnoliales. But in just 1 week, we have 1.2 million individual leaf measurements for more than 100,000 individual specimens. And that's just a taste of what's to come as we keep scaling this up. Yeah, and I think the official term for what I'm experiencing is data delirium. I have more data than I know what to do with now. Yeah, and those 1.2 million leaves is 10 times bigger than any other leaf study to date, and that's just what we can do in 1 week. So another really exciting thing about all of these machine learning methods is just the scale of data collection is insane. And we have a lot of work to do just figuring out the questions to ask when we have access to this much data. So anyway, I also wanted to touch on some smaller use cases for tools like Leaf Machine. Small, but no less useful. We're always looking for ways to extend our machine learning methods to other traits and other tasks. So for example, I've been working with Dr. Joey Charbonneau, who's a postdoc in my lab, and we've been creating tools to measure things like prickles, spines, and thorns from herbarium images. And then we can fit curves to describe all of those shapes. We can also use the archival and plant detection algorithms for seemingly trivial tasks, but they end up saving a lot of time from a collection standpoint. So our fungarium has images like these on the left, where there's a lot of black space, and they would prefer getting rid of all that black space. So we can simply use our cropping algorithms and then create a nice image like you see on the right. Similarly, a lot of other researchers want to use the whole specimen for classification tasks. So things like species prediction, species determination. But using whole images introduces a lot of confounding elements. Machine learning algorithms love to look for patterns in images, and herbarium sheets are filled with patterns that we probably don't want the machine learning algorithm picking up on if we care about the actual plant. So for example, if you try to train an algorithm for species identification, and all of your images for a single species happen to come from the same herbarium, it will start identifying the label text and not the actual plant. So one thing we can do with Leaf Machine 2 is simply censor out all of the text. So this significantly unifies the training data, and you end up getting a machine learning algorithm that works much better. And then the fun thing is, once you do this and you apply it to a novel data set in the real world, you don't actually have to censor it anymore because it's only trained to find plant differences. So that's a happy benefit. There's also some analyses that we end up doing. Those are things that we can do with a lot of different component detection algorithms. But one thing that I've spent far too much time working on is what we can do with archival components. So every single image has a scale bar. And in this case, we've developed quite a few computer vision and machine learning algorithms to convert pixel distances between all of these unit markers into metric distances so that our measurements can actually be useful. And in this case, it's a little bit hard to see, but all of the cyan and dark green points are individual dots that our algorithms have found. So this is actually taking the geometric mean of more than 1,000 data points. So when this works, it is by far the most accurate way of getting a conversion factor when you can't control the imaging step. And if you've seen me give a talk about Leaf Machine before, you know I have a love-hate relationship with herbarium specimens, because this is just a small sample of the rulers we've had to deal with. There is no standardization for herbarium specimens, and it's actually surprising how often there are no rulers at all in some of these images, even from large institutions. That seems to be a relic of early 2000s or 90s digitization efforts. But I'm here to say that some rulers are better than others. These two prove to be the most reliable. So all I ask is that if you're in charge of imaging, please keep machine readability in mind. We don't know what these images are going to be used for in the future, but these sorts of rulers, it's a lot easier to make computer vision models to deal with something that's this high contrast, that doesn't mix metric and imperial units together. And having more units is not always a good thing. There's plenty of sheets that have 3 or 4 rulers in them, and that actually just complicates things unnecessarily. So you can see just based on this spread, these 2 rulers are just much more consistent. So finally, we can talk about what we do with the label text. So every herbarium specimen has an accompanying label, and that's probably the most important thing about this sheet. If you don't have the label, you just have a leaf on a sheet. You have nothing, no other context. So from these labels, right now, if you're in an herbarium, we have to manually transcribe all of these, and you can imagine that's a very expensive process. We can look at a few labels to also see that this is not an easy task sometimes. If you have a modern label, it's been typed, it's very easy to understand. But if you go all the way back to 1880, just reading their handwriting is a problem, let alone figuring out what Formosa is, you know? So geographies that have changed multiple times in the last century. So this on its own, we have a huge diversity of input labels. For current manual transcription processes, the curator has to transcribe the label and then put it in the correct field. And a human could manage between 15 and 20 transcriptions per hour if everything's going smoothly, maybe a little more if your data set is like something like this from 2014. And most of this time is spent cross-referencing external data sources, performing a Google search to figure out what the geography actually is. And then also just the time it takes to type out long paragraphs of text. So anything we can do to speed up this efficiency would be great. With older labels, it might be even worse. So sometimes I can't even read what's in those labels, but we need to transcribe it. So the efficiency slows down a lot if you're dealing with older specimens. And to put all of this in context, only about 48 million specimens have been digitized globally, but there's at least 330 million specimens that have yet to be imaged or databased. And that's just what we know about. There are plenty of collections that they don't even know how many specimens they have. So if we do some napkin math, it will take decades before all of these have been digitized and databased. Any tool we can create to reduce this backlog by increasing efficiency and throughput, by reducing costs, will be a significant benefit, not just to the institutions, but also to science, because we have no idea what's in these collections in the first place. And that neatly brings us to these three related projects that we've been working on. And it's our transcription tool called Voucher Vision. And this project is beginning to take on a life of its own, which is fantastic. And I think that's a goal that every open source project would like to have. And up front, I wanted to acknowledge these wonderful folks who have helped me get this project off the ground. So James Mickley at Oregon State University has done a huge amount of effort helping us stress test our methods. And then Jason Best at the Botanical Research Institute of Texas and Ryan Allen at the University of Colorado Boulder have also given us a lot of feedback. And then finally, the folks at Royal Botanic Gardens Kew. We've started working with Kate Gill, John Adcock, Nikki Nicholson, Sarah Phillips, Benjamin Bowles, Kane Lindsay, for helping push Voucher Vision to the next stage and figuring out how we can actually scale this up to the institutional level. And the goal of Voucher Vision is essentially to use optical character recognition to first read the text and then use large language models to organize messy, unstructured text into a structured format. So in our case, that would be a JSON dictionary, which is easily turned into a simple spreadsheet. So this is just a basic overview of what we're actually trying to do. We start with the full specimen image. We use Leaf Machine 2 to crop out just the text to create a label collage image. And then the label collage is submitted to an OCR engine so we can actually get the text back. So now that we have our raw unstructured text, we need to interface with a large language model. And to do that, we need a prompt. So for Voucher Vision, we assemble image-specific prompts with 4 main sections typically. And this prompt is just the text that we give to the large language model to instruct it to do a particular task. The instruction contains some basic instructions that guide the large language model, including some specific formatting instructions. And these instructions are based on the exact same instructions we give to our transcription staff who would be doing this manually. So as part of our prompt, we have some initial instructions, and then we show it the unformatted, unstructured OCR text. We show it an example of what we want it to produce on the other end, which is a JSON object. And even a year ago, this step was hugely problematic. Fortunately, most large language models are starting to be trained more and more to output JSON text. So this problem is sort of solving itself. And then finally, we provide granular rules for each column where you can specify specific formatting, like putting the date in a specific format. We then submit this prompt to our desired large language model, such as OpenAI's GPT-4o, Google's Gemini, or any of the other open source large language models. And the large language model will return a JSON candidate object. And then we have some additional logic that makes sure that we're returning a JSON object that's valid and that has all of the fields that we require. If it fails, we pass the JSON object straight back into the large language model with instructions to correct it. And usually, if it fails the first time, it'll get it right the second time. So after Voucher Vision processes a batch of images, we send it to manual review. So we're still, at least in our museum, not trusting it to just put the record into Symbiota or Specify or any of those data management portals. So this is just the whole thing, all as one big step. We start with the full image and then go all the way to getting just a spreadsheet where each institution can figure out what they want to do with. What I just described is one of 4 different implementations that we've been working on in Voucher Vision. What I described can be referred to as prompt engineering, where the prompt text contains all of the relevant context and instructions required to complete the task. And for Voucher Vision, we've tested many different implementations of prompt engineering. So we've started with really short prompts, really long prompts, and also combining different OCR engines, and sometimes just showing it 2 copies of the exact same OCR data gives you better results. And that's due to the fact that machine learning models need to pay attention to information. So sometimes just showing it twice literally gets it to pay attention to it more. It's pretty weird. Yeah. But there are other approaches for this. A second approach that we've been working on a lot lately is fine-tuning our own large language models. So in this case, this approach is much more efficient because we basically, instead of relying on a very large prompt that might be 2,000 words long, all we do is create synthetic optical character recognition data from an existing Darwin Core record. So you take an existing row in a spreadsheet and you take it out of the spreadsheet and you just turn it into a string. That becomes training data for a machine learning network to basically do everything in one step. So instead of providing it a big instruction on how to reformat it, you just fine tune it to do only that one task, which is pretty fun. This method is a lot more efficient, but it also gives you some restrictions. If you start wanting to transcribe 25 different fields, you're stuck with that unless you want to retrain a new model. But it's also possible to train some of these in 4 or 5 hours. So it's not the end of the world if you do need to train a new one. And one thing we've been talking about with Kew Gardens is possibly training many of these models on purpose. So if you have a specific collection, you could tailor certain models to certain collections. So a third approach that we've implemented is also pretty cool, and it uses AI agents. So you have multiple large language models working together to solve the same problem. You have a supervisor agent that delegates tasks to worker large language models, and then the supervisor does some quality control checks. And when this works, it's pretty fascinating. It can correct typos. If the state of Colorado was placed in the country category, it can undo that. So to some extent, these are self-correcting, which is pretty cool. But the downside is they're also more expensive and they take longer to work. Instead of just maybe 10 or 20 seconds, this could take a few minutes. So this might not also be suitable for every application. And then finally, we have the version that I think will win out in the long run, which is doing all of this in one step. So even just in the last few weeks, this has become even more achievable. So in this case, all we're doing is sending the image to the large language model. We skip the OCR step because a lot of these large language models have become multimodal. So you can send the image and the instructions at the same time, and it does everything all at once. And when you try to wrap your head around that, that's pretty weird. That means it's actually reading the text in the image, figuring out what everything is, putting it in the right category, seemingly all at once. And it can do other things like converting units. So if you want everything in meters, it can convert feet at the same time. So these algorithms are just wild, trying to figure out how they do all of that. But this will probably win out in the long run as these vision language models become more and more capable, specifically at handwriting recognition. 2 weeks ago, there's a new model that came out called Gwen2 VL, and it does better at reading handwriting than any other OCR method I've ever seen. And it's only going to get better from there. So it's pretty exciting. And I think this method will end up winning out. This is an example of its handwriting ability. This isn't a crazy example, but if you scrutinize it, I couldn't find a single mistake in this. I instructed it to put a ton of information in occurrence remarks. But other than that, I was blown away by this. And this is one more complicated example. And again, it got it completely right, which is not what we would have seen using just Google Vision API or Tesseract or any of the other traditional OCR algorithms. So then there's just a few pros and cons to think about for all of this. And I'm guilty of adding features way too much. So there's a lot to think about. when dealing with something like voucher vision. There's basically two categories of how you can implement this. You can run everything locally on your own hardware, or you can pay an API service. So if you use ChatGPT, OpenAI's models, you'd be paying for an API. If you used something like Llama, you'd probably be running that on your own hardware. And there's just different pros and cons. Local hardware has a high upfront investment. You need to buy a computer capable of running that. But the API has long-term costs, and you can't control the models. OpenAI updates their models every couple months, and it sometimes does impact our performance, usually worse, which is kind of strange, but sometimes the performance gets worse. So that's something to keep in mind, is we have no control over the hosted API models. Yeah, that's just some of the cons I talked about. APIs also give you the advantage of using, theoretically, the most state-of-the-art, best performing large language models, but they can also be incredibly expensive. They seem to be getting cheaper, but if you're not careful, using the all-in-one step we talked about could be $1 a specimen if you aren't controlling what's happening. So this is our vision long-term. We're hoping to support all 4 of these versions, and different institutions might have different requirements. If your collection only has 10,000 specimens, it's probably worth it just to pay OpenAI, use theirs, and just be done with it. If you have 1 million specimens, it's probably worth it to do everything in-house and host it locally. And then finally, I wanted to mention the Specimen Label Transcription Project, which is our effort to sort of bring all of these data into a centralized location. Not everyone's going to want to use voucher vision to do this transcription process, which is great. We need 1 million different ways of doing all of these tasks. But we can all benefit from having access to each other's training data or validation data sets. So a couple institutions have started contributing to this, but if you're interested, I'm happy to talk more about that. It's completely open. I'm not even necessarily the one managing it. I can make you an admin of it. This is supposed to be as open source as it gets, and I'm happy to add anyone who wants to put data in this. So that's all well and good, but we also wanted voucher vision to be easy to use. Some institutions, they might be running this on a cluster. They have an IT person who's comfortable doing that. But we also have a lot of smaller institutions that have a single curator. So we wanted to make sure that we covered our bases. We have a graphical user interface that, based on feedback, keeps getting more and more usable. We also allow our users to customize their prompts, which is pretty exciting. So if you use the prompt engineering strategy, you can customize the fields that you want to transcribe. And we've been exploring this with a lot of different data sets. We've been working with the USDA Wood Collection, the Center for Wood Anatomy Research, to develop prompts for their collection. They also use PDFs, so our models now support dealing with PDF input data. I also helped one of my colleagues, Shelley Gaynor at the University of Florida, develop a prompt specifically for extracting chromosome count from a really special collection that we have. And I've also been working a little bit with the Field Museum's mammal collection to see if we can apply this to mammal tags. And at the end of the day, the answer is if there's text on a label, this or a similar solution will work. There's probably 50 different ways to get this to work, and all we're doing is trying to figure out the way that's the most flexible and most accurate method. Yeah. And you're welcome to try out Voucher Vision. We have a Hugging Face space that's available for you to try. If you go to this link, you can upload an image and see what happens. You could even use our prompt builder, which you can see in the bottom middle, and try creating your own prompt to get really specific information out. So earlier I mentioned that we also don't trust these transcriptions without a manual review process. So we've been working to develop our own editing software. And it's not something that every institution is going to want to use, but in-house, we are going to be using it. For manual transcription, most of the sources of error are typos, so a misspelling of a word, something like that. When you're using large language models to transcribe and you have a manual review step, the most dangerous error is that someone doesn't see text and it gets added to the wrong column. So either hallucination or a date that goes in the wrong field. It's really easy to just accidentally accept something into the official record. So this is a little bit out of date. We keep changing our editing script, but basically we now have two columns. The left column where this map is replaced by the text that comes out of the large language model. And you have to accept it into the record for every single field. So that helps us minimize and reduce the chance that we get incorrect data into the official record. So separating the official record and the large language model transcription a little bit more. We also are trying to include some helpful features in our editor. Right now, this is one of our GPS validation tools. And basically it just shows you if the GPS coordinate is valid. It's pretty common for OCR to miss a negative point or a period. And if you just see that the point is on the wrong hemisphere, you can just, fix that pretty easily. So on that note, I wanted to acknowledge all of our partner institutions again. And while we've primarily been working with Herbaria, this should apply to most specimens and most labels. So if you'd like to get more involved, let me know. And then these are some of the links to all of our projects. If you go to one, you can probably find your way to the others. And then I just also want to acknowledge a lot of our herbarium staff for also helping us do all of this. So thank you.
Yeah, thanks a lot, William, for this excellent talk. And I'm sure there are some questions.
Amazing work.
You can wait for the mic, please.
Hello. This work is incredible. Thank you so much for that presentation. I want to hear a little bit about the process of developing Leaf Machine, because you come from an undergraduate background in biology, right? So you're not a computer scientist. So having tried to do some of this stuff from scratch myself, I'm floored by this. Can you talk about the process by which you got into this and
Yes.
what kind of help you had and what you had to figure out yourself? I'd love to hear about that journey.
Sure. I was secretly an applied math major before. So that's where I learned to code. And what got me started was building an image compression algorithm from scratch. That got me interested in images as a thing. And then everything just spiraled from there. I started in MATLAB, believe it or not. The first version of Leaf Machine was MATLAB, which is fine for prototyping, but is horrible if you want to make something like that. I think starting now is actually a lot easier than like 5 or 6 years ago. A lot of the algorithms we end up using are very commonly used. So even in the Leaf Machine 2 paper, we point out that almost every single image segmentation task for biology uses the same algorithm. So all of us use Detectron 2 or MaskRCNN. So there's a few, how should we say, like MaskRCNN is an architecture, but some companies and GitHub repos have made it easy to use. So like for segmentation, Detectron 2 is managed by Meta. They want everybody to use it. It's very easy to get involved with that. For image annotation, that's another picky step. A lot of people use open source image things like LabelMe or something like that. I use Labelbox, which is proprietary, but they have academic licenses, and we will never come close to using up what they give us access to. But I found that easy. We have, I don't even know, 50 people in Labelbox. All of the data is in the cloud. I can programmatically access it, stuff like that. But yeah, I hate to say it, but Reddit is really good at getting you to the right place first. So what I struggled with at the beginning is trying to like make my own architecture or something. It's a waste of time for us to do that.
Okay.
Companies like Meta, they have people who got their whole PhDs in specifically that machine learning thing. They're going to be better than whatever we can do. So I think our trick is figuring out how to deal with the data side. For Leaf Machine 2, we went out of our way to try to include as many herbaria as possible. Our data, I think we have 300 individual herbaria represented in it. So it should be pretty generalizable. If you're training your own and it's just on like field museum specimens, there's a chance it might not work with someone else's. So if you need it to be generalizable, I would say focus on that. So I would say prioritize data and then try to find some internet resource that gives you the shortcut to whatever model is going to be the most applicable.
Okay.
Yeah.
Thank you.
Yeah.
Hi, thanks for the awesome talk. So obviously this methodology is extremely cool and the amount of data you're getting is so much fun. I'm like
Yeah.
vicariously thrilled for you. But I don't know if you have particular plans to get back to sort of the start of your talk, which was really looking at, how these traits are evolving over time, what pressures are involved, what pressures are you interested in? So I'm wondering if you're working on any of that right now. And also like for those, obviously you have the trait data and you get a lot of the natural history drivers from the labels. Is any of that missing? Do you have to look to other sources or can you get a lot of it just from the labels themselves?
Great question. So yes, I'm working on what to actually do with the leaf machine data from an evolutionary point of view. And that's hopefully going to be a manuscript this calendar year, if everything goes well. But I guess, like I mentioned, I came to grad school wanting to know the macroevolutionary patterns of this. I personally am pretty skeptical of assigning adaptive explanations to leaf shape. I think leaf shape is something charismatic, so we see it a lot. It can be adaptive, don't get me wrong, but I think at a macroevolutionary level, there's some pretty interesting things that probably happened deeper in the phylogeny. And the only way we can get at that is if we can look at a large portion of angiosperms. So with Leaf Machine, we're starting with those two orders and then just going crazy from there. But at a fundamental level, we have no idea the amount of variation at each of these scales. So we have a data set where we measured every leaf on a tree. And it was actually 5 trees, which is crazy. But my bet is that the amount of variation at each of these scales will be pretty upsetting to paleobotany folks. So one of my committee members is a paleobotanist. And yeah, there might be some things that we might have to reconsider from looking at some trends that we might have thought were comfortable, but there's way more variation than we thought there was.
Okay, if there are no further questions, then I want to thank our speaker again and you for attending the talk. Thank you.
Hi there. Oh, thank you. Thank you.
Okay, yeah, let's do like that.